मैम वॉइस नहीं आ रहा वॉइस
Good afternoon, dear participants. Once again, I, Dr. Shagufta Khan, on behalf of the Institute of Pharmaceutical Education and Research, Warda, welcome you all to the second session of the first day program of the AICT-sponsored STTP. Today, we are pleased to welcome a very special guest and an amazing academician, Dr. Sanjay Jajak. Dr. Sanjay Jajak is professor and head of Department of Natural Products, the National Institute of Pharmaceutical Education and Research, NIPER. Dr. Jajak's career has taken him through 25 years of teaching and research. And in teaching and research, he has to his credits grants worth 70 million Indian rupees, the extramural grants from CSIR, Department of Ayush, and DBT, Department Government of India. He has more than 105 published articles in the international journals, and his H index is 35, and total citations amounts to about 3,709. His research experience and area include discovery of anti-inflammatory natural products employing COX-2 microsomal prostaglandin E synthase 1 cytokines as targets, discovery of natural product inhibitors of bacterial efflux pumps, discovery of anti-diabetic and anti-obesity natural products, medicinal chemistry and synthesis of natural products, phytopharmaceutical and nutraceutical functional food development, application of novel drug delivery systems, that is phytosomes, nanoparticles, SMEDs, to herbals and herbal bioactives, standardization of herbal drugs, formulations, and phytochemical analysis. Dear participants, it's a matter of pride to all of us that Dr. Jachak has been featured in world's top 2% scientist ranking published by Stanford University, USA in November 2020. He has been ranked in the field of research, medicinal and biomolecular chemistry, and his rank in India is 28 and world rank is 1270. He has also been awarded Best PhD Thesis Award as Research Guide in May 2017 in Pharma Innova Award, Ahmedabad. And he is the recipient of most cited paper award, that is bioorganic medicinal chemistry letters 2005 to 2008, and he received research fellowship Austrian Academic Exchange Service, Vienna, Austria from 1995 to 1998. He has done PhD in the Institute of Pharmaceutical Sciences, Karl Franz University, Austria. He has guided more than 87 master students, and 14 students have done PhD successfully under him. He has developed syllabus for Master of Science course in Traditional Medicine in 2006, and the course was started from the academic year 2007 at NIPER. And he has developed and updated 2 PhD courses and taught them to PhD students since 2002. In addition to the professor and head of department, he was also associate dean in NIPER from 2011 to 2012 and in charge a small and medium pharmaceutical industry center at NIPER from 2010 to 2011. He was member joint entrance examination JEE of NIPER in 2015 and 2017. He He was member of statutory committees of the Institute, such as Senate and Board of Studies and Research. He was course coordinator, technical courses sponsored by Indian Technical Economic Corporation, Special Commonwealth African Assistance Program, Ministry of External Affairs, Government of India, from 2013 till date. And he was responsible for organization of several courses under ITEC during 2013 to 2019, and he was involved in imparting training to 400 participants from 52 different ITEC countries. And he was also convener international conference drug discovery from natural products and traditional medicines, organized by the Department of Natural Products during November 2018. So Dr. Jajak will share his expert opinion on innovative teaching and learning skills in natural products curriculum. So I request all the participants to give him due attention and join me welcome Dr. Jajak for this session. I request Dr. Sanjay Jajak sir to start with his talk.
Okay, first of all, thank you, Dr. Shagufa Khan, for giving so elaborate introduction. At the outset, also, I would like to thank Dr. Bhushan, then Dr. Ganjiwale, who is Principal Director of IPER Varda, for inviting me to this particular short-term training program at the IPER Varda, and I had been to IPER Varda. So I think long back, 2009 or 10, when Dr. Yevle was actually director principal, he invited me there. So it's very nice institution. So with that, I start with my today's topic. I have chosen natural products as I work in this field. But before that, as I'm saying, the natural product curriculum, there are certain common points that I would like to discuss about the teaching and learning methodologies. So with that, am I allowed to share my screen? Let me see that.
Yes, sir.
Yeah. So
you can start your screen share.
Yeah. So is it visible? Actually?
Yes, sir.
Yeah. So the title, as already mentioned, innovative teaching and learning skills in natural products curriculum. So I will be first half of my talk will be on this teaching and learning skills. Also, friends, we know QBD, quality by design. So that approach, whether we can apply that in education, little bit of that I will also be discussing before I tell about some curriculum in natural product and how it can be research-based teaching can be done to the students, so that they get this discipline or they get interest into this field and deliver the best of their abilities. So with this, I'm just starting first with the elements of innovative teaching, which you will see the general point as all we see, and many of points what I have mentioned are directly related to the classroom teaching, which we are now not doing due to pandemic since last now nearly 9 to 10 months. All of us, we are actually delivering lecture, listening to lecture as we are doing today through Zoom. Several platforms we are using, but in a way, actually, I feel this is also an opportunity, because this has told us, even if the students are not present in classrooms, we can effectively do the teaching by using these teaching aids and the virtual mode. And by virtue of that, I would say we are able to listen to many international people for whom or to whom we will not able to invite personally due to various reasons. And now this is the opportunity we can involve several international scientists, speakers, professors, academicians, industrialists in this teaching mode to the students. And I think the feedback I have got from my students, PhD as well as masters exclusively, they are very happy the way the teaching is going on, but it has its own disadvantages also at the same time. But see, this is an unique opportunity when we are talking about the innovative teaching. So virtual mode teaching itself is an innovative teaching. in a way. So that I would like to tell to all the faculty colleagues who are attending this particular short-term training program organized by IPER. So with this, let us say these are the elements, research-based curricula, student-centered, it should be inclusivity of diversity, as we have diverse cultures in India. Definitely, this is important that we consider the ethnic diversity of the students that we are getting, like at National Level Institute, we get the students from throughout India. So we really see that diversity of students when we teach them. Then the flipped classroom teaching, another concept that is being used under this particular heading, innovative teaching and cooperative learning. So one by one, these 5 points I will be discussing in brief. Like the first one, research-based curricula. As I already mentioned, at NIPER, we adopt this. Whatever the concepts are told to the students, we try to explain them with the research findings and with our own research experiences. I think that gives very good idea and practical insights to the students, so that it is not merely bookish learning or just learning theory to the students. So this is very important aspect I consider. Research-based teaching and the curriculum you develop according to the based on real practical things. Only explaining the NMR instrument theoretically will not help, because I myself I have not understood when it was taught first when I was in third year BPharm. Then I had to ask our teacher to explain again several times. But whenever I entered in MPharm and PhD, I could see it is very interesting. Practically, you can understand many things when it is pictorially shown or demonstration is given, merely teaching on the slide how the NMR works. So that is really the thing, actually, the research-based aspects we need to add into our teachings. As all we are now speaking and interacting to it, with each other, all we are teachers actually. So this is very important, I would like to convey. Then the writing the research grants, projects. This is another aspect that we are talking about. The research projects, we should try to, let us say, motivate students to write small grants, especially I'm talking about PG and PhD student, not the undergraduate student. Help develop and test students' subject understanding, the research projects. Then students also learn to communicate with their research findings to an academic audience, for example, through academic posters and reports during conferences and seminars. Then the next point is student-centered. The student-centered education means giving students responsibility for their own learning. They become independent thinkers by developing the strategies. And the confidence to learn by discovery. Then implement a modular structure that enables the students to have greater choice and flexibility within their discipline. Then develop novel ways to work in partnership with students, like co-create innovative teaching practices, employ them as teaching assistants in classroom. Actually, this is done in the Western world. When I did my PhD, this happens actually. The postdocs and the PhD students, along with the professor who is lecturing, sometimes they bring in to sit and assist the professor in their teaching. So that really gives idea to the postgraduate students, especially that they are involved in teaching along with their mentor. Then inclusivity of the diversity. So creating equal opportunities for students to succeed regardless of their gender, sexual orientation, ethnicity, etc. Recognizing and harnessing students' diverse cultural backgrounds, then identities and experiences, by creating opportunities for them to learn from each other and to make their different backgrounds an asset. So this is also important. Then designing a diverse range of teaching, learning, and assessment approaches that recognize the support, the needs of students, both as individuals and as members of learning community. Then removing the barriers to learning, this is very important, and creating a more equitable experience to assist all students regardless of language support needs. Then the flipped classroom teaching, what it is actually, it is designing lessons such that the student learns content of the lecture, listening to the lectures at home also, as we are now doing, teaching to the students through virtual mode. Then this methodology, it allows the ability to meet with the students one-on-one more often and at a higher rate. the ability to have more group projects, problem-based learning, and more labs. In addition, students are expected to go home to complete their assignment by using the knowledge they have gained in class through hands-on training and lab work projects. This is also can be very well implemented to the B.Pharm students, as I believe firmly this can be taken care by the teachers. This is what actually before the student prepare to participate in class activities. So actually they prepare means they do the homework first for the topic which will be taught in the class. Then during the class, student practice applying key concept with feedback. And then actually in class further, student check their understanding and extend their learning. That is after the class. And this is actually the flipped classroom teaching that we follow. Then the cooperative learning. So cooperative learning involves positive interdependence, individual accountability, group processing, interpersonal and social skill, promotive interaction. So all these are the components of this cooperative learning among the students you can say discussion or with the approval of the teacher. So these innovative teaching learning methodologies, technology enhanced learning, mind maps and flow charts. So technology enhanced learning here. So these digital and online innovations can be used to support and enhance more interactive ways of teaching. Online and digital technology can replace lectures and classroom time, can be used for interactive education experiences. This is what all we are going through. Again, we are discussing the virtual classrooms and teaching is going on. Use of technology can further enhance our education, including real-time, two-way feedback. Then use of blended learning can be used to free up time in the classroom for more interactivity. As the student prepare earlier on the topic, they will be actually more interactive in the class than only listening to the teacher. So this type of experience is very good, and more and more we should try to adopt this particular methodology, that we try to make the class as interactive as possible. So students are given chance to interact. If somebody has some other idea, clarification, or some problem in understanding the concept, can be solved effectively by this particular method. And then student can be asked to study online materials, which are now plenty available before coming to the class, and will be better prepared for the actual interactive classroom teaching. And it also allows to create international classrooms by mixing students with peers in other parts of the world online. And this is what I already mentioned. Now we have the opportunity to invite with no cost all our international colleagues, experts, to give their insights and impart knowledge, share their experiences with the students. So a mind map is a diagram used to visually organize information. That is drawing information in diagrams instead of writing in sentences. Then mind map can be drawn by hand, either as rough notes during a lecture, meeting, or a planning session. Then this pictorial method record knowledge in such easier way. Mind maps are much quicker to make and much easier to remember and review because of their visual quality as compared to then writing paragraphs and pages and pages. So it is better that these mind maps are made like this one here. It is showing the possible mechanism of action of antibiotics as antibacterials, as we teach them in second and third year in medicinal chemistry in B.Pharm level also, and advanced in masters also. So here this kind of actually map one can prepare for different types of antibiotics, as it is mentioned, based on their mechanism of action is already you can say pictorially presented, that actually easy to remember for the students. So I will not go into that. All you know, you are teaching, most of you, the medicinal chemistry, antibiotics, and antibiotics definitely I consider is a class of natural products already. Then while after all these 5 points of teaching and learning, I will tell something about the institution. I am not against any private institution or anything, but there are few points I think all of you, and even if needed, it can be included for the betterment of many of the private institutions which are really doing very good. I know most of them like MIT or many other institutions, I know they are doing very well in northern part of India. I am not in much contact with South, but the northern India, we have so many great institutions, private institutions. They have already inculcated and introduced very innovative teaching learning programs based on research as well. This is important to mention before I discuss about some private institutions as a whole in general without naming anybody. So please excuse me for that. If I say something, it will be my personal thought, opinion. It is not of anybody's thought. So that is what I would like to make it clear once I discuss few slides in the forthcoming presentation here. So while the number of institutions have grown out of proportions, as all we know, the following in particular have not grown proportionately. Quantity of quality teachers and training tools, and hence quality of student training is hampered, and accordingly the quality of research output is also hampered. That is what we see in the last 2 decades, especially when there is a mushrooming of several institutions, not under pharmacy, but also in engineering and technology and ITs, what we see. So pharmacy education today is thoroughly commercialized, as all of you will agree with me, and it is in the hands of market forces also. So the private sector, which accounted for about 10% of the students admitted in 80s, now accounts for 91% of all pharmacy students admitted. So you can see the involvement of private sector in pharmacy education. So as per the Yashpal, Professor Yashpal, who was ex-chairman UGC, he actually opined that our university system has lost its primacy and is now beset by cross commercialization. Its autonomy has been eroded, and actually undergrad education is undermined. And most of you, I think, colleagues who are attending this workshop, you are in undergraduate education as teachers. So this is very important to note. Our autonomy has been eroded, and the undergraduate education is undermined. So that we need to really look into and try to improve, so that we develop better scientists than we are generating today in our country. So very well, this applies today to pharmacy education. That is my opinion. Actually, the undergraduate education is undermined. When I see why I'm seeing this, because I am seeing there is a gradual decline in the quality as well as interest of the students that are being admitted in my department since 2013 or so. I really see there is a lot of downfall. I don't know what are the reasons. There are so many reasons, but this is what students not having concept. They have never done any practical. If I ask them how to extract and isolate rutin, a flavonoid glycoside, from, let us say, onion leaves, so students are unable to tell. They have never done this such type of practical, or even they have not done any extraction also. At several places, the students, they are getting admission to master's program in natural products, I am quoting. So this is where I want to say this applies today to pharmacy education. If this is a situation in IPER, you can imagine what can be there in several other various good institutions in government setup or in private setup. So overemphasis and spending on buildings by managements of private institutes, but less on education quality. So I'm very sorry to say again, but it might be true for very few institutions. Now everyone is up to the job and everyone is doing best as per their ability. Now type of institutions on quality scale. Now you know NIRF rankings are there. So NIRF rankings are based on the output, academic output, as well as several other factors listed out. So in that case also there is emphasis how the training is being imparted to the student, practical training, whether it is research based or not. So with that, I would like to say that in back in 2011, Innovation Award NIPER SAS Nagar received from Thomson Reuters. So what we have highly qualified faculty who are mostly got their education abroad, most of them or all of them, I would say. And interdisciplinary training is being imparted to the student. Best of the facilities we have under one roof and most favored by students. So if you see this actually perception as one of the major points in this actually teaching. So and even NIRF rankings. So we get 100% perception ranking always since the NIRF under pharmacy also started. So this is what actually on quality scale we try to build up. And I urge many of you also try to do the best possible in your best of the capacity. Then the old leading institutions, old and established leading institutions here, major contributors to the growth of profession, scope of further upgradation facilities, other institutions of respectable quality, upgradation can convert them to leading institutions, and local institutions that only meet basic AICTE or PCI requirements. They are here listed, but I now see AICTE also has now moved out from pharmacy. Now it is mostly the PCI who has to take the baton further and try to improve the curriculum at the BPharm level in most of the institutions who are actually who have to comply to PCI norms. So good pharmaceutical companies are avoiding unskilled pharmacy graduates, and that has opened up a path for postgraduate students and have started preferring students from also basic sciences. Like you know, in many pharma industries, we have in quality control department, whenever I have worked, and I think it applies to today also, that the MSc people are there, then more MSc than more MPharms in the quality control department of a pharma industry. So those things are coming in. So basically a BPharm student has been rendered a cheap labor as a cheap labor, and so is going to happen soon with the postgraduates, with the MPharms. So this is a really alarming situation for all of us to handle. The problem is going to become more acute in the future if there is further increase in seats by AICTE without proper planning and consultation. Now, as I'm speaking today, AICTE has a lesser role, but still it is there. So one has to see actually that whatever the increase in number of seats has to be rationalized by AICTE by consulting with the institution, and there should be more interactive sessions before granting this increasing seat by the institution in question as well as the regulator like AICTE or regulatory body there in education. Unfortunately, in India, we have a dual statutory system which is going out of control. This is another problem because, as you know, the PCI is not under any other ministry, but it is under Health Ministry. The Pharmacy Council of India is under Health Ministry, whereas you can see the regulation is also under Health Ministry. But if we see IPERs, we are not under Health Ministry. We are under Ministry of Chemicals and Fertilizers, where there is a separate department created in 2008, Department of Pharmaceuticals. So now presently we are 7 IPERs. So all 7 IPERs are under Ministry of under Department of Pharmaceuticals, Ministry of Chemicals and Fertilizers. Then the Pharmaceutical Pricing Authority is also under our ministry. So there is actually already the pharmacy education is divided. It is not under one statutory body, and this is really a problem when we see many of time. So the uncontrolled growth of institutions further is likely to become steady as those established with sole objective of making quick buck will close, and under the influence of market forces, the rule of survival of the fittest will eventually prevail. So not much worry on this account. The uncontrolled growth of institution is already now controlled. So there is not much worry in this aspect of the increasing number of institutions. Then the unfortunate situation that a pharmacy graduate is treated as cheap labor mentioned earlier by employers must be amended. And here academic institutions are expected to a big role in this direction, because they should be trained, they should be made more knowledgeable when they go for the interviews. Like everyone is giving emphasis nowadays on placement. So depending on the placement percentage ratio, the quality of the institution is decided. So that's why academic institutions are expected to play a big role in this direction, so that BPharm students get better placement, and we encourage more and more BPharm students to take post-graduation, and they will not stop only after doing BPharm, but they can also go for the post-graduation in pharmaceutical sciences. Change in motto of academic institutions from excellent building to excellent teaching and training infrastructure with greater emphasis on modern instruments, attracting and hiring best faculty as much as possible, continuous high quality faculty development programs, which are, I think, going on a lot more, as I see a lot of QIPs are going on currently in virtual mode, as well as this type of STPs and STTPs are also going on. I think in the last 3 months, I have given several lectures in such programs, including the today's one, as I am speaking to all of you. So excellent academic buildings are beautiful only if they are well equipped from inside also. So like you have the best of the instrumentation, as these are the pictures from industry. Okay, these are not from academia. Whatever you see inside, these are the quality control department of pharma industry. But we would like to see that such things also in our colleges. Okay, so and anyway, this below one, this is PXRD, and this is the actually NMR instrument, all that which are there only in the higher education institutions and R&D institutions you find. But like to have the HPLC demonstration to the students, GC demonstration to the students, arranging some practicals, still that is definitely needed at the BPharm level itself nowadays. And this is what actually I mean. They look beautiful when they are equipped from inside also. So the teaching shall be concept oriented and not only bookish, I already mentioned, as followed by most of the pharmacy teachers. So we should try to see that it should be more concept and research based than only have the theoretical base. And curriculum must be interdisciplinary in line with global trend. Because as I believe I'm working in natural product, it really gives me a lot of opportunity to do interdisciplinary research. Like I can do, let us say, the formulation development, which is otherwise everyone say a pharmaceutics student will do. But why not a natural product student can do the same thing? So you try to do the research, and also in times try to collaborate, like for certain advancement. You collaborate with the pharmaceutics person, or for that matter, I want to know mechanism of action of some compound, natural product compound that shows activity in vitro. I can collaborate with good biologist, good pharmacologist for understanding its mechanism of action, and so on. So you can actually see so many different areas I have written. In the heart or in the center, it is pharmaceutical sciences, and I believe pharmaceutical sciences is the applied science, is the applied chemistry, basically applied sciences. That is what actually we see in pharma. So we need to also have the interdisciplinary approach with the basic sciences, and with that mind, I think all NIPERs have been, especially NIPER Mohali, the first NIPER has been designed. We have actually 10 departments with 14 courses at master's level, which are very much interdisciplinary at NIPER Mohali, I can tell you that. And same model was followed by all other new NIPERs that were created under the same ministry. So curriculum can be as possible as you can make it interdisciplinary as much possible as there as you can, and that will really increase the knowledge level or enhance the knowledge level of the students. So also it must be designed with the futuristic vision, involving all the stakeholders. So pharmaceutical education in India, central government policy, approval of establishment of central universities, national institutes, etc. Statutory bodies like AICTE, PCI, etc. Education regulation, institutional approvals, course curriculum, updating this practitioner registration, student fellowships, etc. by these bodies. State governments, again, approval of for the establishment of state universities, local institutes, then UGC, DST, DBT, CSIR. These are, and many more. These are the research funding organizations under Government of India. And they provide the research grants as well as fellowships to the students to pursue their masters as well as PhDs. Then industry, CROs, CRMs, hospitals, clinics, medicine outlets, CDSCO, state drug control departments, government and private laboratories, etc. They actually provide a lot of employment in private as well as in government setup for our students. And professional bodies, as we have several in India, like IPER, IDMA, APTI, and so on. And students actually who are the receiver, who are the main focal point in all this exercise, they are receiver of education. So in that background, now, as I mentioned earlier in the beginning, can we apply this concept, which is not new? People think it is a new concept. I don't think it is a new concept. It was very much there, but that was there in certain engineering fields, not in pharmacy. That's the only difference. So in aeronautical industry, if there is no QBD, you will see many, you can say, the accidents with the aeroplanes. So QBD and automobile industry. So where they were using this since last 6 decades, this QBD. Okay, so that has been now adopted in pharma. So on that, you can say, background, I would just like to ask the question whether is it possible that we extend this QBD to pharmacy education, and answer is definitely yes, as we will experience through the next few slides. So QBD is a new concept in pharma, applicable to pharmaceutical development and production. So many of you would have read this dedicated issue in August 2014, dedicated to actually QBD, Pharma Times. And many more, I think, several then magazines, journals in India, they have published a lot many articles on the adopting the QBD in the manufacturing setup in industries. So you will find many articles now already on QBD in pharma. So it is rather a revolutionary change for assurance of product quality. That is the main goal of applying QBD. So quality by testing is now replaced by quality by design, QBD to QBD now. And QBT, that is quality control by inspection and testing of finished product, is now overtaken by QBD, quality built into the finished product. No reliance on end testing as it is there in QBT. That was the approach, I think, post or before 2010, pre-2010 or so. Now, I think many companies in India, last 1 decade, they have made enormous efforts to apply the principles of QBD. And risk analysis. That is what QBD is. One of the principles under QBD is also risk assessment. Risk analysis is also part of that. And many pharma industries have adopted now this approach. So when we say that, we should have the process analytical tools. So PAT control at every step of processing is now possible. So there is no emphasis you have to give on the end testing, as earlier it was done. When I was in industry, that time it was done. So here now, online particle size analysis, like it is mentioned, raw material testing, dispensing, dry compaction, wet granulation, FBD milling, blending. So this is the, let us say, capsule and tablet actually, or let's say here the capsule formulation is being mentioned, and even the bottle filling, the liquid orals. So you can use several techniques during these processes are going on, like the near infrared spectroscopy. So we can use that in material identification, then vision particle size analyzer, then NIR, again, conformance testing for the raw material testing. Then for these processes, online particle size analysis when we are making granules. Then we can use also NIR for determining the loss on drying, online UV cleaning, online blend uniformity when the process of blending is going on. Then NIR can also be used for checking the core potency during compression. Again, NIR has a lot of applications. In chemical imaging method, we can use in capsule filling and so on. So you can see the near infrared spectroscopy has enormous application as one of the tool process analytical tool that will be used, that will ensure or build the quality in the product, so that there are less chances of its failure once you will have the final product or final formulation. So that is the role of this PAT in the QBD. So these are the key components of or key elements of quality by design. Define target product quality profile, design product and manufacturing process, identify critical quality attributes, process parameters and source of variability. And lastly, the fourth one, control manufacturing processes to produce consistent quality over time. So this is what the concept is, QBD, that has three spaces: knowledge space, design space, and control space. So in design space, we use a lot of DOE, design of experimentation. As all you know, you must have studied, many of you also might be working, applying this QBD design in formulation development. Then now, after telling this concept, how we can apply this QBD to education. So extension of QBD to education, that is the today's topic, innovative learning and teaching. So in that, you see QBT, quality by testing in education, to be converted to quality by design in education. So quality by testing means having the examinations at the end of the semester or at the end of the year. Whereas now we are talking about quality by design in education. Let us see QBT, quality through testing and examinations, as I mentioned. Whereas QBD will have building quality in students through effective design policies, curriculum, and teaching training program, incorporating continuous evaluation of the students. Like we do assignments, we give surprise test. Okay, we take the oral vivas. We tell the student to prepare short seminars and prepare. So this is actually going on, continuous evaluation through different modes that we can do. And that can be actually called as QBDE, quality by design in education. So expected teaching quality, young students want solid education where curriculum and teaching methods are up to date. And that is what we can use such methodologies to enhance the interest of students in teaching, in learning, and as well as in research. As I already mentioned some time back, that I see there is downfall in the students' quality. So we cannot only say the problem, we need to also find a solution to that. So the solution could be, yes, definitely, we can do the continuous evaluation of the student, make it more actually, you can say, as much as possible objective than writing only subjective questions and answers. So all those things we can try to adapt, and even that is there in PCI curriculum also, that they notified the B.Pharm curriculum as well as M.Pharm curriculum, as PCI has already made it mandatory to follow by all the pharmacy institutions across the country. So they want teaching and training, students want teaching and training that helps them adapt to reality of 21st century and is useful for entering active life, the world of work and society, which is becoming extremely competitive day by day, as all you will agree with me. So therefore, the objective of teaching and training shall not be just increasing the key qualification of the students. And as it is practically happening with mushrooming of colleges and uncontrolled increase in number of seats in pharmacy institutions. So where only degrees are distributed with minimum emphasis on knowledge and skill development. So QBD, a teaching and training program that aims at all students of a class having similar knowledge and skill set as assessed through a continuous evaluation program, just I have mentioned. And despite that, students of a class, maybe with different backgrounds, levels of intelligence and capacity to learn, the students are given certificates of completion of the course and degree at the convocation. So end examinations not necessary or given very low weightage due to continuous evaluation that one can consider doing, like mentioned here. So earlier final exams at the end of the year, 100% weightage was given. Whereas now two summative assessment, like this is from the, let us say, CCE and CBSE pattern. Now SA1 year-long evaluation, SA2 final exam. So final exam has only 30% weightage versus 70% is given to the continuous evaluation year long. So this is what need of an hour. We need to do more and more such things so that student can learn more, and also they can learn, they can enjoy the learning while they are in the classrooms or virtual mode. So a great advantages of this particular approach can be freedom from anxiety and stress from tests and examinations, as we all have gone through that, as we know what kind of anxiety and stress brings to the students when they appear for examinations like board examinations and so on, and many joint entrance examinations that are held in the country. So this is a photograph from Bangkok University. Make students wear anti-cheating helmets during midterm examinations. So which you can imagine extremely or an extremely stressful situation the student can experience when writing the examination. So such type of things are also there certain countries, so that should be avoided. Then as such also in today's environment of campus placements, the employers hardly pay attention to mark sheet, and it's very true. So these paper sheets have no relevance left, but as a qualification we do need. But basically the across the table, the industry personnel actually test the knowledge of the student, knowledge level, than looking at its percentages wholly. So they give a lot of emphasis on the knowledge part and less emphasis on the percentage of marks, for example. So those kind of things are there, and across the table when you take the interview, immediately you come to know the knowledge level of the student, starting with its actually with the aptitude and the body language of the student, how he sits or how he or she presents. The person across the table or on another side of the table can make out what is the knowledge level, and then they test that. Instead of asking them theoretical questions, the practical questions are being asked. So that is what we need to actually train the students, more practical based, research based, and what is going on currently in the current scenario, than whatever was done in 70s has no relevance today. So we have to tell them all the current advancement in a particular field, in a subject that we are teaching, so that they are more knowledgeable and they are better prepared for the interviews when they go for placements, and of course communication skills. So this is very important. Like most of the pharmacy students we get, they are from rural background, very poor in communication skill, and I must say very poor in English and grammar. That is what I see at IPER Mohali also, but I don't know what you people see that, but this is my experience there. Then the expected QBDE elements in now education. So at macro level, these are the ones. These are the 4 key elements of QBD, and there actually you will see with respect to education, what is that particular at macro level. You see those elements like pharmacy student with knowledge and skill suitable for the job. That is, you define the target product quality profile, the design product and manufacturing. So robust education policies, high quality institutions, infrastructure, curriculum and faculty. Then identify the CQS. So CQS, good employability for each and every student. Then the control manufacturing process, control type and total number of institutions, quality of faculty and student intake number. So this could be 4 elements of QBDE when you see at the macro level. At micro level, you can see the first one here, target product quality profile, a student whom any employer would hire without a blink. Then robust, when you say about design of product and manufacturing process, robust institutional teaching and training program, CQS, equal learning to all the students, and identify all the variables that will improve the knowledge level of the students. Control on all variables through efficient mechanisms. So these are at the micro level, QBDE. And the steps that you have to take: build knowledge space, assess expectations and annual job vacancies in industries, CRO, CRMs, CDSCO, and other regulatory government and private testing laboratories, hospitals, modern chain of pharmacies, etc. Study education and research model of leading Indian and international institutions. List all variables that can influence equal training of students in a class. So that is nothing but building a knowledge space, as we are talking about 3 spaces in QBD approach. So knowledge space, build design space, use optimal statistics to assess the requirement of the number of institutions, infrastructure, faculty and student, use best statistical and design tools for selection of the right education model, use design models to outline most suited curricula, apply design of experiments, DOEs, to identify the variables which require stringent control. And the control space, controls that can be implemented by regulatory agencies and also educational institutes to radically change the current scenario of pharmacy education. And this is what the burden we say on the students, that you can't help. Burden will be more and more as they go to higher studies anyway. So, but this is, I think, a very old quote I'm putting, but that is interesting to know, as expressed by a student on Facebook. समंदर जितना syllabus है, नदी जितना पढ़ पाते हैं. This is very true. बाल्टी जितना याद होता है, गिलास भर लिख पाते हैं. चुल्लू भर नंबर आते हैं, उसी में डूबकर मर जाते हैं. Yeah, this is the actually story of most of the average and mediocre student. If you see in a class of, let's say, 60 or 100, so we need to really see and assess the knowledge of students so that they are not feeling as it is expressed by one student on the Facebook, for example. So it is a scientific tool for making education and learning a fun, and that is the great challenge ahead of us or in front of us as a teacher that we see. And this is what I'm just showing now after all these preliminary things about the teaching and learning methodologies applying QBD to education. Now, as I'm teaching to natural product student, I have just compared already a few years back the M-Pharma curriculum as actually in pharmacognosy notified by PCI in semester 1, semester 2, like having what kind of subjects, what is the depth of those subjects that is being covered in the course structure that was seen, and accordingly what is the practical lab. As you see here, in the first semester hardly we have any practical, so that has gone into the research methodologies, biostatistics later on. Okay, and then you will see the research work, journal club, and research work. So whereas we do give a lot of emphasis on practicals in the first semester itself, as they have to complete 3 credits of different practicals, lab hours. So we have very quite exhaustive in the afternoons. We have only lab throughout the week. Okay, we have 20 weeks semester, 10th semester is a midterm examination, and lot of assignments, seminars, and surprise mock tests are given to the students during let us say a semester, so that we try to see it is more continuous learning. And even seminars are there which will carry the credit. And you see in the next prior semester also we have all the advancement in the particular discipline, as well as a lot of emphasis on the general lab experience, and the projects are given to the students in the first semester itself for the 3rd and 4th semester, where they have to do the dissertation work. So that is what actually our structure in MS Pharm syllabi from IPER Mohali I am giving. And this is another course that I had developed, traditional medicine, in 2006, where also we have given also emphasis, lot of emphasis on practicals and research-based learning, and also the hospital setup, clinical setup to the traditional medicine students by taking the pharmacy practice courses. So with that, another important aspect when you go for the innovative teaching, use a flow chart. So more and more flow chart. Flow chart is a type of diagram that represent an algorithm, workflow, or process illustrating a solution to a given problem. And the steps in a flow chart are shown in different shapes depending on its purpose. It will vary from subject to subject. They are connected with an arrow, and flow charts are used in analyzing, designing, documenting, or managing a process. Like here, all you know, flow chart of the typical HPLC instrument. Like if you want to tell the student theoretically, you first tell them this flow chart and then show them the instrument. So they will understand it is nothing but a simple instrument that can be very easy to handle and use. So that is what this pictorial type of learning teaching will definitely help students to understand better than just telling them one by one and just describing its function. So this is what the typical flow chart of HPLC instrument, analytical HPLC instrument for that matter. And with that, I'm just showing one or two examples. If I have 10 minutes, I will just give two examples from my lab done by one by the PhD student, one by the master student. Just an example. There could be several, but with the time limitation, just this plant we are working for some time. Try also develop the herbal formulation out of this by doing the detailed study. As all you know, it is known as Punarnava or Rakta Punarnava in Ayurveda. So this is Borrelia diffusa. Whole plant is shown here in the natural habitat and the roots are used. So these are the traditional medicine uses. As one of the research focus in my labs is actually the discovery of anti-inflammatory natural products. So we have taken, keeping in mind this plant for the anti-inflammatory research application of this particular Ayurvedic herb, Borrelia diffusa. So those uses are mentioned in traditional medicine, for example, in Ayurveda. Then this is a pharmacopoeial status of B. diffusa. We do see a monograph in 2007, 10 and 14 IP on this plant in Indian Pharmacopoeia. And it tells Punarnava contains not less than 0.005% weight by weight of boravinon B calculated on dried basis. So boravinon B is considered as a phytochemical reference standard or a marker for the chemical standardization as well as assay for the roots of this plant, which are used in Ayurveda for different therapeutic purposes, including treatment of inflammatory disorders. And these are the, when we have started work, we have checked at the phytochemistry in the roots as well as aerial parts. So what we observed, these particular 10 rotenoids, which are modified isoflavonoids, that have been reported from the roots of this plant. So accordingly, the plant material was collected, voucher specimen, that is all. And very important part that we have prepared, rotenoid rich fraction. Enriched fraction, not from the 70% whole ethanol, 70% ethanolic extract, which is a whole, and that was simplified into a rotenoid rich fraction. And that I will explain with the chart like this, like the 20-gram extract is taken, which is made by maceration, 70% ethanol extract that is subjected to column chromatography on diane HP20 resin that was eluted with water methanol in gradient manner, starting with 100% water, reaching up to 100% methanol. And what we see, 90% methanol in water actually, and 100% methanol fraction concentrated on rotary evaporator. They were pulled together based on their TLC profile and concentrated on rotary evaporator. That gave us rotenoid rich fraction 1.1 gram from 20 gram. You will see a lot of interfering compounds, some sugar materials are there, and other compounds like you will see some a little bit of the pigments, though they were very less, but the polar components like sugars and certain disaccharides or trisaccharides were there more in the roots. Those all were removed and we got the compounds of interest. And this is actually practically done, shown to the student. And then actually he optimized this process further and increased this, as is the first trial, the yield towards 2 gram. So we started with 1 gram and end up in 2 gram by optimizing the process here, that preparing this rotenoid rich fraction from the whole extract that was prepared from the roots of this plant. So this is what we need to prepare chart, and I use this actually also to the teaching to the students during practicals and also during the classes, for example. So this is the HPLC profile. and then we have done the LCMS in order to identify the compounds. So based on the molecular weight that is obtained, information and UV pattern from LCUVMS, we could identify known as well as unknown compounds, and unknown compounds found to be a new natural products that were identified for the first time from the roots of this plant. And this was actually the detailed work done by the PhD student. He had isolated around 12 rotenoids, 10 are mentioned here, including the marker that was mentioned, boravinon B or phytochemical reference standard. And it is actually the major compound. As you will see here by the area under peak, this is the major compound, that is boravinon B among all as the marker for this plant, but its percentage is not less than 0.005. So you can see they are present in very low percentages, but still among them, this was the major used as marker for standardization as well as for assay of this plant. So that was, I will not go into details about the isolation, characterization, and then actually identifying and new compounds. So those are mentioned with the code numbers were found to be new compounds. And this is once we did that, we have done the large scale isolation. This was done first on actually 2 kilogram scale, then we have scaled up to 8 kg, and all these compounds were isolated in let us say 10 to 100 milligram quantities. Otherwise they were obtained less than 10 milligram initially. So that is what when you deal with natural products, you generally end up in isolating very less amount. So this was scaled, and then boravinon B even we could isolate in 500 milligram quantity when we started with 8 kg of the roots of Borrelia diffusa. That is a major compound, and this is the flow chart. That is how actually I give this example to our master students, give them this practical also, which was already published, done by the PhD student during their actually first semester and second semester practical classes. So this is what the detailed schemes flow charts are being made and given to the students, and also given the training in the structure elucidation and interpretation of the spectral data, so that you determine the structures of the new compounds. And this was all the biological experiment. I will not go into detail. And this is the work done by my master student. Very good work. He had isolated 5 compounds from the stem bark. All we know, very high valued medicinal plant in Ayurveda as cardiotonic. So here the stem bark is taken, and these are the compounds. So the student had isolated all these 4 and this glycoside from the, let us say, out of the 6-7 compounds mentioned here. The student had isolated, master student, 5 compounds. Out of that, you will see in the literature, people write arginolic acid, and they give the structure actually as this compound. But what we find, arginolic acid as a marker is actually not a single compound. It is the structural isomer of 2, arginolic acid and argonic acid, as you can see there. Even we have only a difference of hydroxyl group extra in this at this particular position. That is, if you see the 20th position in this pentacyclic triterpene skeleton, if you see there. So it is a kind of Woolian skeleton that we see, and you see these are the having both the compounds having same molecular weight, arginolic acid or argonic acid. That's why they are structural isomers. Like this OH group, from here it is shifted to here, CH2OH we have here actually, for example, and from here it is shifted to here. That is only, that's why I said these are structural isomer. In the literature, this is already described as argonic acid. Wrong actually, structure determination and report that we have clarified, and this is under publication currently from Terminalia Arjuna. So this is the example, like the student is given this. Actually, they have studied different methodologies, how to extract and isolate the compounds, making flow charts in practicals during first and second semester. Could do this within 1 year, all this study and characterizing these compounds. So that Terminalia Arjuna bark, now there is already a monograph on this bark also in IP 2010-14 So terminally Arjuna contains not less than 0.02% of arginine calculated on the dried basis. Because they have put arginine, because this is difficult to isolate these two compounds. So this is arginine, one more related structure with additional hydroxyl group. So you have 3 here. In this case, you have 4. So that is relatively easy to get. That's why I think the Pharmacopoeia Commission and laboratory have included arginine as the marker for terminally Arjuna. But earlier in the literature, they have given this compound as arginolic acid as a marker, which structure is wrong. It is actually argonic acid, for example. So these are the things when you go into that, and you see the difference. The student has prepared triterpenoid enriched fraction from this, let us say, ethyl acetate extract, and you see all these phenolic compounds which are absorbed at 254 nanometer are found to be absent here. Like you see 256 nanometer here, there is no peak, which are actually the UV absorbing compounds. And triterpenoids are, they are like you have seen, devoid of strong chromophore, except this double bond in the compound. You don't have, and the carboxylic acid. So they are not having the chromophore to be detected by UV. So we have detected that at 210. So here you see many peaks. So that is what it is, a triterpenoid enriched fraction, as I have shown you, the rotenoid enriched fraction. So this is what is required, current trend in natural products. Simply making extract will not serve the purpose. We need to prepare enriched fraction, and where actually the methodology of the phytopharmaceutical. As all you know, this is the first time in the world India has introduced, our CDSCO and DCGI has introduced in 2015 new category of drug called as phytopharmaceutical. And there is a requirement in phytopharmaceutical, you have to prepare enriched fraction that is qualitatively and quantitatively standardized and estimated with at least 4 phytoconstituents. So this is what, if you adopt this approach, you can very well go for the, let us say, translational research from your laboratory to the product. If you adopt this approach, and that will enable you to go for phytopharmaceutical product development. That is what I need. I want to convey message to all the faculty who are working in medicinal chemistry or pharmacognosy. You can very well take such type of researches, if feasible in your laboratories, can be actually have a lot of commercial applications. So I think with this, I thank you all. This is our glimpse of the entrance of our institute, those who have already visited. And those who have not visited, actually, I invite you to visit sometime once the pandemic is over, from next year onwards. We hope so that it will be over. And then this is our natural product field laboratory, which is near the main gate. And we are very fortunate and lucky to have 20 acres of land on IPER campus, Mohali campus, for the medicinal plant garden and cultivation of the medicinal plants of interest. Okay, and these are the buildings. So I'm here in the C block. I'm sitting here. Here is my room. I'm sitting and talking to all of you. So here is the HOD chamber, wherein we are having at the first floor, second floor is a natural product, ground floor we have pharmaceutical analysis. So a lot of analysis in this block, whether the phytochemical or pharmaceutical we do here. And this is actually the other teaching and research blocks that you see. This is our main area, teaching and research block. So with this, again, I thank you all for your patient hearing and paying attention to my lecture. So I will be happy to answer the queries you have. I think I have finished almost in 1 hour and 15 minutes. So I hope we have some 5 minutes at least for the question answers. So thank you so much.
Dear participants, the session is now open for question answer session. If anyone is having any kind of questions or queries, they can put it into chat box, or they can ask by voice also. Sir, there is a question in chat box.
Okay.
By Dr. Kiran Erande, sir.
Okay.
He's saying the concept of design, space, QBD, PAT, HACCP, etc. are really appreciated and accepted by industries. But is it feasible for academic institutes to accept and implement QBD in education? As for many private institutes, it is tough to follow PCI norms.
Okay, so that is what I have mentioned. Yeah, as per PCI norm, it is not there in the syllabus. I know you are right there. But as we are looking for, let us say, the more and more innovative and advancement in teaching, so why not to accept this? This is what we actually, as we are not under PCI, so we can adopt. I know you can say that, but yes, yeah, that is how the experts can tell to the PCI Executive Council, or to the wherever you have a chance or a platform to convey this. Actually, PCI will definitely would like to consider, because Professor Saraf is very much actually open on this, and I think we had one forum already last year, when before the pandemic, in some private institution in Punjab. I think somewhere. I think next to Ludhiana, we were in an institution. So where Professor Saraf, we had this. So they are open. Actually, they said if more and more suggestion comes from different institution, we will consider. Actually, that is what I can say. It is a future. And according to my personal views, it is important that if you make it more practical based, research based, it will be good for the students. Like many students have not seen some instrument, okay, IR. So suddenly when they come in MPharm, then it is problematic to them. Theory they have read, studied, but when it comes to the practical, let us say for that matter, preparing KBR pellet to record the IR spectrum, student feel it very difficult. So that's why, if I think as far as it is feasible, institutions can include this, and PCI can also be written. I think many institutions are doing. Like I know Amity is very big group, but Noida I have visited. IPC was there 2 years back in 18. So I have seen this actually. Very good infrastructure under private. All institutions are not that lucky, I know. But yes, it can be followed. Why not?
Thank you, sir.
Yeah.
Any other questions from participants? Okay, I think there are no more questions, sir.
Yeah, thank you. So
on behalf of IPER Varda, myself Abhiram Deshmukh would like to express my deep sense of gratitude to you, sir, for your excellent coverage of teaching techniques. Your talk, which covered key elements of QBD, syllabus of research-based learning, and interdisciplinary teaching and its importance in the overall development of students apart from bookish knowledge, were phenomenal. Your concern about the declining quality of students is indeed a cause of concern for all of us. And certainly we received a lot of motivation from your thought and views. I thank you, sir, from the bottom of my heart for taking out time from your busy schedule and sharing your views. Indeed, it was a great pleasure to share a moment with your esteemed personality. Thank you very much, sir, for being with us today. Thanks again. Thanks a lot, sir.
thank you all, and I hope every one of you have enjoyed and find it useful in your day-to-day teaching process, because all we are teachers who are interacting, listening to each other. Yeah. So thank you so much.
Thanks a lot, sir. Have a great day.
Thank you. Yeah.
Dear participant, tomorrow's session will start at 11 a.m. So we will join at 10:50 a.m. with the guest speakers. The feedback link is already posted in your chat box, so you can fill the feedback form till 8 p.m. Thank you very much.
Thank you.